pleasure to introduce Dr. Lisa Levin. Lisa obtained her bachelor's at Radcliffe College, but I believe that she actually became interested in marine organisms at the age of 16 when she went to do a special marine organism course at Humboldt University. And I've nearly broken her computer. I have to be careful what buttons I press here. Sorry, Lisa. She then obtained her PhD right here at Scripps Institution of Oceanography at UCSD. I think we'll pass very hastily over the fact that she did a postdoc at that place on the East Coast. For those of you who don't know, that's Woods Hole. She then went to North Carolina State, and then she came back to Scripps Institution of Oceanography as a professor in the Interdisciplinary Oceanography Division, IOD. And she has more than 20 years' experience with shallow and deep-sea marine environments and the animal communities that live there. And at first glance, I think it may seem a little strange that she studies both communities of animals in really shallow water and also in really deep ocean. But as Lisa says herself, she's one of those people who never stop playing in the mud. And of course, there are sediments at any water depth. And so Lisa and her team of scientists and students explore the ecology of invertebrate communities inhabiting muddy sediments in ecosystems ranging from the deep sea to intertidal salt marshes. Now, she says that one of the reasons that she loves oceanography as a career is that she gets to travel to remote parts of the world and has friends around the world, and that makes almost daily discoveries that are so exciting for her. But I think the thing that really impressed me most is that who else do you know who can say Peru, Chile, Mexico, Oman, Pakistan, Alaska? Those are just the few regions whose sea floors I have come to love. So, at least that impressed me no end. We're very lucky to have such a distinguished ecologist and oceanographer here tonight, and I have actually been waiting all day for this talk. Deep ocean is a world that fascinates me so much. It remains such a very distant and remote place that we know so little about. But tonight we're going to get an opportunity to learn a little bit more about the bizarre populations of organisms that manage to thrive under really the harshest conditions in the deep ocean. So with that, Lisa, please come forward and tell us more.
Can you hear me? Yep. Okay. I'd like to thank Nigella and others for inviting me here tonight, and thank you all for coming. What I hope to do this evening is to describe to you two environments in the deep sea that you probably don't know very much about. They aren't very well known, and these are oxygen minimum zones and methane seeps. And these are two of the most stressful or harshest of the deep sea environments. But before I do that, I want to take a step back, sort of bring everybody up to the same level in knowing what the rest of the deep sea is like so that you can place these environments in context. And I'm going to start with the definition of the deep sea, because there are, in fact, many definitions. And for most people, the deep sea would be anything deeper than one can scuba dive in. And so that might be, I don't know, 30 meters or maybe something like that. I don't know how deep people scuba dive. But for most scientists, our definition of the deep sea would be beyond the shelf slope break. And I think many of you know the continental margin typically has a shape like this, where there's an abrupt decline at the edge of the shelf at around 200 meters. And so for many people, that's the beginning of the deep sea. And for the really hardcore scientists, the deep sea begins at 1,000 meters water depth, or this would be about 3,000 feet. However, many of our submersibles, manned submersibles, only go to about 800 or 900 meters, and so they stretch that hardcore definition a little bit. But in any event, there's a lot of the deep sea out there. We know that 70% of the Earth's surface is covered with water, and most of that is, in fact, in the deep sea. So what is the deep sea like? It's cold, and the average temperature, except in the Mediterranean, is between about 2 and 4 degrees centigrade. It's very dark. We know that there's no sunlight down there, but there is some light because some of the organisms bioluminesce and make their own light. It's salty. The salinity is very constant, around 34.8 parts per thousand, and the pressure is high. I'll come back to pressure in just a minute. And there's not very much food that makes its way from surface waters down into the deep sea. Now, to give you some sense of how things have changed, prior to 1850, scientists, and everybody pretty much, thought there was absolutely no life at the bottom of the deep sea floor. That idea was retracted basically in 1859, when a communications cable was hauled up from the Mediterranean and totally encrusted with organisms. And at that time, scientists realized those animals were living down there. There must be life in the deep sea. This set off a series of expeditions. One of the first and most famous is the Challenger expedition, which explored water depths greater than 2,000 meters for about 4 years. And in truth, much of what we know about the form of the deep sea and the kinds of organisms that live there were discovered back in the 1870s on the Challenger expedition. There were a whole series of deep sea explorations that took place after the Challenger, with some hiatus during the World Wars, but they continued on up through about the 1950s when the Galathea expedition occurred. This was really the last great trip in the classic expedition sense, and on the Galathea they studied really deep depths, the hadal depths, greater than 4,000 meters. They did the first microbiological studies and the first quantitative abyssal samples at 4,000 meters and below. So at that point, the big expedition stopped, but hypothesis testing continued and much more focused research cruises took place. And then relatively recently, scientists discovered hydrothermal vents in the 70s, and then in the 80s, cold seeps. And I'll be talking about cold seeps tonight. I think most of you have heard about the discovery of hydrothermal vents. I put them on this list because I think these are some of the most important recent findings in the deep sea. Well, from the early work, up until about 20 or 30 years ago, scientists believed that the deep sea was a very stable, quiescent, food-poor environment, that organisms experienced the same conditions all their lives, and that these, in fact, didn't change from generation to generation for hundreds, thousands, or possibly even millions of years. But over the last 20 years, a series of different really greater access to the deep sea and a series of discoveries pretty much changed that old paradigm. And now we recognize that the deep sea is very dynamic with respect to the input of food, kinetic energy, and that species abundances actually change seasonally sometimes and interannually as well. We know that there are deep sea storms that occur in some places, and we know that there are huge pulses of food. A dead phytoplankton that we call phytodetritus rains down from the surface to the sea floor, sometimes seasonally in many parts of the ocean. Now, the stresses experienced in the deep sea, I've listed 3 of them up here: pressure, oxygen, and sulfide. And by far, pressure is the best studied of the 3. Pressure will increase 1 atmosphere for every 10 meters of water depth, and that means on a 4,000 meter deep sea floor, the organisms would be experiencing 400 atmospheres of pressure, whereas we're sitting here today experiencing 1 atmosphere. So there's tremendous pressure, but I'm not going to say any more about pressure because most deep sea research into stress is in fact on pressure. I will talk about oxygen and sulfide. It turns out that much of the deep sea is pretty well oxygenated, but there are regions where oxygen is in short supply, and that's what I'll be talking about. And also sulfide. Most of the deep sea floor doesn't have much, but there are particular environments, including methane seeps, that have very high concentrations, and sulfide is quite toxic. So let me start with oxygen. Now, I have to tell you, about a week and a half ago, or 2 weeks ago, I heard somebody give a talk and they used music. And I was so fascinated with that approach that I thought I'd try it tonight. And so in terms of, so you'll hear a few songs, if I can get my cursor to show up. And what can we say about oxygen? Well, the Hollies and the Bee Gees said it very well. Okay, so we all need air. We all need air to breathe, and it's true of deep sea animals as well. It's essential. Can't live without it. Oxygen in the ocean comes from a variety of sources. The plants in the ocean, the phytoplankton, will photosynthesize. A lot of the oxygen in the ocean comes by mixing of waves at the water's surface. And also, new seawater formed at in the North Atlantic is very high in oxygen, and it sinks and spreads out through the rest of the world oceans. And so those are some of the key sources. We know that much of the ocean water is saturated with concentrations of 5 to 7 mils per liter, but sometimes the oxygen can be very low, and when it's low we call this hypoxia. It can be defined in many ways, often just less than 2 milliliters per liter in concentration. Now, most of us are familiar with hypoxia, or low oxygen, in its man-made form. This figure shows the distribution of man-made hypoxia, and you'll see it's focused around population centers, mostly in the northern hemisphere. And this is the result of excessive input of nutrients, sewage, different kinds of organic pollutants that use up all the oxygen in coastal waters. However, most people don't recognize that there's a tremendous amount of natural hypoxia in the ocean, and it has almost the opposite distribution to the man-made hypoxia. There are layers of water in the ocean that we call the oxygen minimum zone, where oxygen is very low, less than 0.5 mil per liter, and this occurs mainly in the eastern Pacific Ocean, off West Africa, and in the northern Indian Ocean. These oxygen minimum zones are formed by 3 basic processes. The most important of these is upwelling. These are upwelling zones which yield very high phytoplankton production, and as those phytoplankton sink through the water, they begin to degrade, and that uses up oxygen. But that alone is not sufficient to form these zones. It turns out that the waters in these areas are naturally old, and that is because new seawater formed in the North Atlantic makes its way slowly through the Atlantic and then spreads through into the Indian and Pacific Ocean, and by the time it gets up to these regions, the water's old, maybe thousands of years old, and it's lost much of its original oxygen. And then the third factor is that these areas also experience sluggish circulation at mid-water depths, and so there is no influshing of oxygen from current systems. Now, this is a plot of the low oxygen zones as a function of latitude and water depth. And so what you can see is that the oxygen minimum zone occurs at different depths in different places in the eastern Pacific Ocean. Off Canada, Oregon, and California, it occurs quite deep, below about 600 meters. But off Mexico and Peru, it's very shallow, and it comes up onto the shelf, and this is true a little bit off Chile as well. The oxygen minimum zone is about 1,000 meters thick off Mexico, so it's probably best developed here of any region in the eastern Pacific. Now, where these mid-water low oxygen bands impinge upon the continental margin, they create hypoxic benthic environments. And many of you may not realize that there's quite a famous song written about the oxygen minimum zone. Let's see if I can get my cursor to show up. There we go. Okay, I'm not sure even the singer knew she was singing about the oxygen minimum zone. Basically, that's what happens. The animals that encounter these low oxygen bands of water experience shortage of oxygen, although I must say many of them have evolved to adapt to these conditions. Now, if you were to view the oxygen minimum zone along the continental margins, it would look like this narrow band of yellow here. This is Baja California and the part of the coast of Mexico. And I've worked with John Helly to calculate, knowing the depths of the oxygen minimum zone in different parts of the world and the topography of seafloor, how much of the seafloor in the ocean is naturally hypoxic. And we've come up with about 1.1 million square kilometers of naturally hypoxic seafloor. So this is quite a large area. However, this is not a permanent area. We know these features do last over tens of thousands of years, but they can also change in some places as a result of climate. And a very dramatic example is what happens off Peru during an El Nino. The 97-98 El Nino off Peru was a very severe one, and it turns out we calculated the oxygen minimum zone area was reduced by 61%. And this is because equatorial waters from further north are flushed into the region, and these actually bring better oxygenated water than the ambient conditions. And as a result, there were all sorts of fisheries, scallops and hake, and a few other kinds of fisheries flourished in these areas, and normally they're either depressed or completely inhibited by the low oxygen. So the El Nino can be a good thing for Peru. Now, these scenes may look familiar to some of you. Periodically, in the La Jolla region, we get mass influxes of animals from the south. Giant squid and tuna crabs are two examples. And we really don't know why they come. Sometimes they come with El Nino, and we know that there is some low oxygen water brought up during the El Nino, but sometimes they don't come with El Nino. But I have a hypothesis that the low oxygen water masses, which are sort of lurk offshore and in slightly deeper water, periodically come up and may be driving animals inshore. But that's just a hypothesis. So I'd like to now turn to the oxygen minimum zone and ask what lives in these low oxygen sediments. And one of the strangest kinds of organisms are these giant sulfur bacteria. They're literally the length of your finger, not quite as thick, but big. And they're called spaghetti bacteria for obvious reasons. And they occur in massive quantities off Peru and Chile, and so one can trawl through the bottom and bring up literally armsful of these things. There are also many, many different groups of worms, especially the annelids, and polychaetes and oligochaetes are present in low oxygen sediments. Now, a lot of my work is focusing on the groups of animals that live in the mud. These are the animals retained on a 0.3 millimeter screen are really what I study, and we call these macrofauna. They include worms and crustaceans and mollusks, all different invertebrate groups. And if one samples through the oxygen minimum zone in the Indian Ocean, off Oman, which I've done, one finds that the stations within this oxygen minimum zone, indicated in red here, these first four, are dominated almost entirely by polychaetes. There really isn't a very big diversity of animals. And it's not until you move into deeper waters where oxygen in fact increases that you begin to see a different suite of invertebrates present. Another feature of the oxygen minimum zone is very low diversity, as I mentioned. If you take samples through the oxygen minimum zone, these solid lines, and into deeper stations, one sees a huge change in diversity. And this is how scientists represent diversity. These are called rarefaction curves. Oh, thank you. Very useful. And what these curves show you is that for a given number of animals collected, how many species you might expect to find. And when you sample the core of the oxygen minimum zone off Oman and collect 100 animals, you might only find 5 or maybe 10 species or 15 species present. But if you move deeper and deeper and collect 100 animals, you might find 25 species or even 50 species present. So there are huge changes in diversity, which we believe are associated, at least in part, with changes in oxygen. Now, how do animals live in this low oxygen system? They have to have some kinds of adaptations, and I'm not a physiologist, so I've only been able to see the most obvious ones. they have hemoglobin. These animals are red. Not only are the worms red, but the sea cucumbers are red, and the mussels are red, and even the tunicates are red. So many animals have red blood made red by hemoglobin to facilitate oxygen uptake. And it's important to realize that in the rest of the ocean, these animals aren't red. If you think about the mussels that you find on the seashore, they're normally cream-colored or even grayish. And same thing with sea cucumbers and some of these other groups. So just the representatives of these groups that live in these very low oxygen waters have evolved to use hemoglobin. Another kind of adaptation I call fuzzy, furry, and feathery. They have very elaborate morphologies that enhance their surface area. Most of these animals take up oxygen through the body wall, and so the higher their surface area to volume ratio, the more oxygen they're going to be able to take up. And I show you just a few examples here. These are amphipod gills. They have all sorts of elaborate feathery gills. These polychaetes all have elaborate cirri and branchiae, sometimes along their sides and sometimes on their heads. And this is one of the strangest ones. This is a nematode. Normally, for those of you who know marine nematodes, they're smooth and sleek and featureless, but these are basically hairy, and it's just speculated that those hairs may facilitate oxygen uptake. And that group of nematodes is only found in oxygen minimum zones. These animals also have an unusual adaptation in that many of them build dwelling structures. The mussels live in mud cocoons, and many of the worms build either mud balls or tubes. And we're not really sure what this is an adaptation to, except that the sediments of the oxygen minimum zone have very high water content. They're almost like soup. Think about pea soup. That would be about the consistency. And it's possible that these structures are just a way to stabilize themselves and keep themselves possibly situated near the sediment surface. Now, my interest in low oxygen systems actually began when I was studying seamounts, independent of oxygen. And I was fortunate enough to visit Volcano 7, about 100 miles offshore of Acapulco. And after working on this volcano with a series of geologists, we noticed there was hardly anything living on the surface. And it was then, I should say on the top, it was at that point that I went back and looked at the hydrography of the area and realized that this seamount stuck up into a very severe oxygen minimum zone. Remember that thick area I showed you off Mexico? And the top of the seamount has less than 1 mil per liter of less than 0.1 mils per liter oxygen, and oxygen increases as you move down the flanks of the volcano. So I'll show you some pictures that really illustrate the dramatic changes we see, probably as a result of oxygen, at least in part from the oxygen. The upper summit is totally barren. There are rocks. They're fuzzy. They probably have a variety of protozoans living on them, but they haven't really been sampled in any detail. And occasionally a mobile animal like a fish will come in and out of the zone. But if you move just off the summit about 50 meters deeper, the oxygen goes up a little bit, and then there's an explosion of life. We have sponges. These are serpulid polychaetes all over the rocks. We have galatheiid crabs. We have huge densities of brittle stars all over the place. And then if you begin to move further down the seamount, in fact, the animals disappear again. The flanks and the base have very few epifaunal animals. We sampled the macrofauna in the sediments. These are the invertebrates, the small invertebrates that live in the sediments, and found the same pattern. Basically, at the very top, almost nothing. Huge densities as you move just a little bit deeper to this zone right here, and then the densities decline again. And what I think is happening is that there's an enormous amount of food coming down through the oxygen minimum zone undegraded, and that is because no zooplankton live in that low oxygen water mass. They don't eat anything, and so it comes down, and low oxygen prevents degradation, actually, of the organic matter, and so it just piles up, and nothing can live there, and nothing eats it. But you get a little more oxygen, and all of a sudden some things can live there, and there's a great food supply for them. And then as you move further down the flanks, the combination of more oxygen And the greater depth, I think, decreased the food supply, and so there are fewer animals there. And this strange pattern, once I began to look, I found it all over the world. These are samples these are transects across oxygen minimum zones of macrofauna off Peru, Africa, Oman, Mexico, Central California. Not on my work, you can see other authors, but in all cases there's a very low density in the core of the oxygen minimum zone, and then the oxygen goes up, usually between about 0.1 and 0.2, sometimes 0.3 mils per liter, and then there's a huge peak of animal abundance. You see it over and over again, and then it goes down again. So this seems to be a global phenomenon. And at those peak abundances, the assemblages can be just incredibly dense. And here's one of my favorite examples off Oman. At 1,000 meters, which is where the oxygen just begins to go up, there were about 100 spider crabs per square meter. Well, these crabs are about yay big. They're not huge, but those densities are just incredible. So there has to be a lot of food for them to eat there. These are some pictures from Volcano 7. But many of the cruises I've been on, I haven't had cameras to document visually the densities, but when we trawl, we take up huge numbers of animals. So what are these animals eating? Well, we know that many of the animals eat phytodetritus, dead phytoplankton that come down. And in fact, when you look at the sediments in some of these places, they're green from the phytoplankton. See how green it looks? And we see flocculent phytodetritus material in our sediment cores. And I've worked with scientists where we've brought cores up on board ship and done feeding experiments with labeled phytoplankton, and sure enough, a lot of animals eat this. But what turned out to be really strange is that there are quite a number of animals that can live in this environment that don't use photosynthetically produced organic matter. They use chemosynthesis as a food source. And they do this through by hosting endosymbiotic bacteria that use chemical energy sources. In most cases, it would be sulfide, and their bacteria would be sulfide oxidizers. So we found this in a very abundant oligochaete worm that lives at 300 meters off Peru. These worms have a whole host of symbiotic bacteria. There are at least 5 kinds, and only 2 of the kinds, the sulfide oxidizers and the sulfate reducers, have been identified, but there are a number of other types of bacteria living under the cuticle, and some researchers are working to try to figure out what they're all doing. And then there are a host of bivalves, leucinid bivalves and thiacerid bivalves that live in these environments and host sulfide oxidizing bacteria. And then there are other animals that eat the free-living spaghetti bacteria as a food source. And so it turns out that there are chemosynthetic and photosynthetic pathways in these low oxygen zones. And prior to this, we really have been thinking only about hydrothermal vents and methane seeps as our main chemosynthetic environments. And I think people need to expand their thinking a little bit. So the message from the study of oxygen minimum zones, one of the important messages is that the animals can live under these incredibly low oxygen conditions and have evolved to do that. And I think we're just beginning to learn how. So now I'd like to move to a different stressful environment, the methane seeps, and start by talking about what is methane. I think most of you know what methane is. It's clear, odorless. It's what we call natural gas. It heats some of our homes and runs our gas stoves. And it's also a greenhouse gas. So as methane increases, it can warm the atmosphere. Now, much of the methane that we know about comes from anoxic sediments or soils on land, such as rice paddies or salt marshes. And we also know that cows can produce methane. And I'd like to share with you a song about this. This is the last song, I promise.
I am cow eating grass, methane gas comes out my and out my muzzle when I belch. Oh, the ozone layer is thinner from the outcome of my dinner. I am cow, I've got gas.
My kids like that song a lot, so I thought I'd share it with you. Okay, so methane comes from all those sources, but it also comes out of the deep sea crust. As the Earth is squeezed through subduction processes and other tectonic processes, methane will sometimes bubble out in what looks very much like a jacuzzi. And so I'm going to play you a short movie here, if I can make it work. Give you a sense for what a methane seep might look like, a very active one. I'll play it one more time because it's a short movie here. You can see methane bubbling out of the seafloor. And this methane can actually fuel specialized animal communities, and these are what we call methane seeps. Now, methane seeps often have very black sediments, such as you see here. These are some of the most toxic sediments on Earth due to very high sulfide concentrations. Sulfide is hydrogen sulfide. It smells like a rotten egg. Any of you who've walked through a swamp or a salt marsh probably know that smell well. But the animals, actually, that live at methane seeps love it, and I've come to love it from studying these systems. These ecosystems actually rely on both methane and sulfide as an energy source for bacteria, and the bacteria fuel the animal communities. So very often where methane is coming out of the seafloor, we will have what we call hotspots of biomass, big animals in big clusters. But of course these aren't really hot like hydrothermal vents are hot. These are cold habitats. They're 2 to 4 degrees centigrade, and so we call them cold seeps. Now, 20 years ago, we believed that hydrothermal vents were the only deep sea environment that relied on chemical energy. And I think most of you are probably familiar with these scenes of hydrothermal vents. But we were proven wrong in 1984 when methane seeps were discovered. Now, the first discovery came from some scientists tootling around the base of Florida here on the seafloor in Alvin, looking for something, and they were geologists. I don't know exactly what they were studying, but they came across zones of tube worms, and then up against the escarpment that is the wall that forms Florida, they found black sediments and big populations of mussels and snails living on the mussels. Now, since that very first discovery in 1984, right over there, you can see that a huge number of methane seeps have been found globally. And these are, in fact, not all of them, but just all the ones that I could find reference to. And I've updated Kathy Campbell's map a little bit, but these are the blue squares. And in fact, once we recognized methane seeps on the deep seafloor, scientists began to better understand that what they'd been looking at in the fossil record, these yellow areas, were also fossilized methane seeps. And they occur over a broad range of depths. The shallowest methane seeps are in very shallow water, and the deepest ones are in trenches. So if these are methane seeps, why does sulfide occur at high concentrations? And it can be very high, up to 20 or sometimes 25 millimolar. And the explanation's a little bit complicated, but I'm going to try to explain it. Now, I'm not a microbiologist, so I've learned all this from reading, really. Basically, the methane fuels a group of microbes called the archaea that are anaerobic methane oxidizers, and these are represented here in the green. And they live in consortia with bacteria that are sulfate reducers, represented here in the red. And the methane comes up, it's basically eaten by the anaerobic methane oxidizers that then produce organic substrates which fuel the sulfate reducing bacteria. And these bacteria take sulfate from seawater, and it comes down into the sediment, and they reduce it and produce hydrogen sulfide. And it is that hydrogen sulfide which then fuels the symbiotic bacteria that live inside the clams and mussels and tube worms that are so abundant in these settings. So what lives at seeps? Well, when you read about seeps, you will find most of the animal descriptions focusing on vesicomyid clams, middled mussels, and vestimentiferan tube worms. There are a few other bivalve groups that also occur there, lucinids and thiocerids, and this Solomyid, this acarax species. And these are all the animals with symbiotic bacteria, and they are the biggest animals at methane seeps. And the reason they're so big in what's normally a food-poor deep sea environment is because they can exploit the chemical energy, the chemical food sources at methane seeps. A few other animals that have symbiotic bacteria include some snails and some sponges. And then there are some other strange animals, some giant aplacophorans that we've just found, and ice worms that live on gas hydrate, which is a solid form of methane that occurs at seeps. And we really don't know yet what these animals are exactly eating. Now, as I mentioned, I love to study the little animals in the mud, and it turns out most of those don't have symbiotic activity. They're what we call heterotrophs. They eat other animals, or they eat mud or bacteria. And there are many, many polychaete groups. I've listed some of them here that are common in methane seep sediments, as well as amphiliscid amphipods. There are also big animals that will scavenge at seeps. So there are shrimp. There are brachyurine crabs, there are giant isopods in some places, hagfishes, and even starfish. So there are all sorts of animals living in these deep sea seep environments over a very broad depth range. And then, last but not least, there are the bacteria. The bacteria often form these white mats that you see. These are sulfide oxidizing bacteria. They're very large. It's thought that the seep bacteria help precipitate carbonate rocks. We often find these big slabs or boulders of carbonates present at seeps. And in some places like the Black Sea, they can create huge edifices, 3 or 4 meters high, that look sort of like giant stalactites and stalagmites. And scientists are just beginning to learn that these structures are really made of bacteria. Seeps are also sites of large intraspecific aggregations of animals, and I think we don't know exactly why yet. This is an amazing picture. These are snails that have come aggregated to lay egg cases in these tower-like structures. The snails each are about that big, those white animals, and the egg cases are about so high, and they've created a huge field of egg cases. The snails are all moribund. They apparently lay their eggs and then die, and the young must hatch out. I don't know too much about their life history. This is off, we've seen these off California and various places. And then there are dense aggregations of sea urchins we've seen off Oregon. Again, we're not too sure what they're eating, but something they're associated with the methane seeps. Now, I'd like to talk a little bit about sulfide and its effect on the animals. And to do this, I want to tell you how we study sulfide in sediments. Due to the miracles of modern technology, there are now needle-thin electrodes that one can introduce into the sediment and profile sediment down core without really disturbing the sediments. And this means we can do other things with those cores, like look at the distribution of animals. And this is my former postdoc, Vipka Zibas, who's done all the electrode work. And when we sample sulfide in different methane seep environments, we find that different habitats that look different on the surface have totally different sulfide concentrations. So for example, the bacterial mats I've shown you have very high sulfide concentrations, up to 10, 15, sometimes 20 millimolar, with sulfide present all the way up to the sediment-water interface. This shows sulfide going down the core into the sediment. Clam beds, on the other hand, have a rather lower sulfide, but still high. It's up to about 2 millimolar off Northern California, with no sulfide present in the upper 4 or 5 centimeters. This is due to their pumping activity, which oxygenates the sediment. And then the non-seep sediments, shown here in green, really don't have much sulfide at all. And it turns out that if we actually section our cores and look at the distribution of animals on the same millimeter scales that we can measure the sulfide, we can plot the abundance of animals against the abundance of sulfide, the concentration of sulfide. And I was amazed to find that all the plots for all the different animal groups we looked at, except 2, showed this L-shaped pattern. This basically reflects the fact that animals hate sulfide. Sulfide binds to cytochrome oxidase and basically can suffocate animals. It can keep them from getting oxygen, so it's toxic under most conditions. And so as soon as the sulfide level goes above about 1 millimolar, the animal densities are 0. They really don't like it. The exceptions were the vesicomyid clams that form these clam beds I've been showing you pictures of, and this small group of polychaetes, the dorvalaid polychaetes, which seem to have very high abundances at from 1 to even 14 or 15 millimolar sulfide. And I'll talk about those more in just a minute. If we look at the diversity of animals that live in the sediments, we find that the most sulfidic environment, the microbial mat systems, have the lowest diversity. Again, these rarefaction curves show that a sample of 100 animals might only yield 10 species, whereas if we sample non-seep or even the clam bed sediments with lower sulfide concentrations, we get many more species. Now, it turns out that the microbial mats are very toxic environments, and most animals are excluded from these. But there is one group, these dorvalaid polychaetes, which are very, very abundant in this setting. And I became interested in them when I began to realize that instead of 1 or 2 species, which is like what I thought we had, it turned out there were many, many species of dorvalaids living in these sulfidic sediments. So my research has, over the last year or two, has sort of begun to focus on questions about how do these animals live in this environment, and are they partitioning the environment? How can multiple species co-occur? We found up to 17 species now in seep sediments in Oregon and California, just of this one little nondescript type of family of worm. And we're interested in, I've teamed up now with a physiologist and a molecular geneticist to ask questions about their evolution and about how they tolerate sulfide. And we should be having some cruises in the next couple of years to actually answer these questions. One of the other ways that we can study sulfide in the deep sea is by putting out experiments. Now, it's not so easy to do experiments in the deep sea, but it can be done. And what we've done is take the large colonization trays with a central cup and put in experimental treatments in these cups. We put the trays down on a gear elevator, and then we use either a submarine or a remotely operated vehicle to come remove the trays and place them into the sediment. And this illustrates what the tray consists of. There's a broad rim and a central cup, and in that cup we put defaunated sediment, and we also put agar with or without sulfide present to add sulfide to the system. And we've done this just once for a 6-month deployment in which we added agar in the lower layer, mimicking the clam bed. Unfortunately, we hadn't seen the profiles for microbial mats, and so the next time I do this experiment, I really want to put in higher sulfide levels up near the sediment surface. But we did put these out both with and without sulfide, and we put these trays inside and outside seeps. We did 6 replicates per treatment, and I'm just going to show you the results for the dorvalaids. We were interested in whether the sulfide attracted any animals or inhibited any animals, and in particular whether the dorvalaids, which tolerate high sulfides, would come in to these trays. And it turns out there were a couple of groups of animals which only came to trays with sulfide added, but by far the biggest effect had to do with where the tray was placed. The trays put inside patches of seep sediment got the highest colonization of dorvalaids. And we don't know if this is because they don't migrate very far, they just came out of the sediment and into our trays, or whether it's because there's some cue emanating from the surrounding sulfide sediments that we got very high colonization in those trays. We're still working on this problem. Another really interesting question is how these tiny little nondescript worms, most of them are less than a centimeter long, most of them are half a centimeter, and there are multiple species living together. How in the world can all these species coexist? Well, if we look, one possibility is that they are partitioning the environment with different diets. And when we look in their guts, we actually see bacteria in one species and sediment in another. And sure enough, their stable isotope values, which reflect their diet, are very, very different, telling us that they're eating different things. So the delta C13 signatures of an animal reflect the signatures of their diets. And if their diets are different, then you would expect to see different numbers. Now, we looked at stable isotope values for only 3 species thus far. We're really just beginning this work. But when we plot them on a dual plot with 2 isotopes, nitrogen delta N15 and delta C13, you can see that the three species sort out very well. So what we've learned is that they're each eating something completely different. These tiny little white worms that all look alike and seem to be in homogeneous sediment are finding different food to eat. And whether it turns out that the other 15 or so species that we have in the system each all have different diets, we don't know yet. So this work has, there's lots of places to go with it. We're very interested in the mechanisms of sulfide tolerance and how these animals actually can live at such high sulfide concentrations. I'm also very interested in the fact that this group seems to have undergone evolutionary release. Those sulfidic sediments are so toxic that most other animal groups normally found in sediments don't live there. And it appears that this one group has come in and multiple species have, I don't know if they've radiated in this environment or just evolved in other environments and come in to take advantage, but it looks like there's some kind of radiation going on, and we don't really know what the speciation mechanisms are, but we're interested in finding out whether different sulfide tolerances are driving this speciation process. And it turns out that dorvalaids don't just occur in methane seeps. They can be very abundant animals in low oxygen minimum zones. They occur on dead whale carcasses, what we call whale falls on the deep sea floor, but they're also very common in polluted settings in shallow water. They occur at pulp mills and sewer outfalls. They're different groups, but in fact the same genus. The genus Ophryotrocha is found commonly in shallow water, and we have 10 species of that genus at the methane seeps as well. So in closing this talk, I'd like to actually take a step back and ask the question, well, why should we care about these really unusual deep sea, stressful deep sea environments, methane seeps and oxygen minimum zones? And sort of from a pure scientific fascination, I would like to suggest that stress is probably one source of biodiversity, that the animals to live in these strange systems have had to evolve novel adaptations. They have very strange morphologies. Some of them appear to have radiated in these systems, and they've proven to us that animals can live almost anywhere in any conditions, some very surprising conditions. From a more anthropogenic point of view, it's quite likely that these environments are going to provide some products and services to people. Now, methane seeps are the site of gas hydrates, which are a solid form of methane. And while they're not currently extracted as an energy source, as our other sources of energy become depleted, it's quite likely that the energy companies are going to be looking at methane. In fact, they already are at methane hydrates. So it remains to be seen if we'll be running our stoves off those. There's the possibility of drugs, unusual enzymes that can function at high sulfide or low oxygen concentrations. Food. I hate to think that these environments are going to be major food sources for us, but certainly oxygen minimum zone boundaries are zones of animal aggregation, and there are dense assemblages of fish. I think some of the fisheries that, as we move into deeper and deeper water, are going to be harvesting from these aggregations. And then, of course, these systems, in particular, the broad oxygen minimum zones, play key roles in nutrient cycling and carbon sequestration, and so ultimately can affect the global climate cycles. But one of the things I have to say is that almost all of these systems that I've been describing occur in bathyal depths on continental slopes. Not in the deep sea, but at sort of intermediate deep sea depths. And these slopes are right adjacent to population centers and human activities. And while I put up here that exploitation of the slopes is imminent, it really is occurring already, and we are fishing on our continental slopes through lots of trawling. We are extracting oil and gas on our slopes and moving deeper and deeper pretty much every week. And we have used for a very long time our slopes for waste disposal, and this is especially true in the less developed countries. So what I'm hoping is that that an understanding of these strange environments will engender some appreciation and put us in sort of a conservation mode so that we can maintain our seep and oxygen minimum zone ecosystems and not turn them into these kind of burying grounds, what we did with deep sea corals that have been really heavily trawled throughout the world. And with that, I will stop and I guess answer questions. Thank you.